Thank you very much. So, pleasure to introduce the next session and the new theme of Applied Child Health Informatics, which Pia Hardlin and I co-lead. And the intention of this theme is really to do a few things. First of all, is to leverage and make better use of all of the data we have through our EPR system. So that's using our digital research environment and our drive unit, is linking up with all of the expertise we have in ICH through PPP. And also an important arm is how we can do the next, if you like, the next level of data with devices, and that's where we're linking up with Sheffield and the NIHR Children and Young People's MedTech Cooperative. So you'll be hearing some examples, and we'll give you a little taster of some of those things. Kate's going to talk about a super important concept, which is how do we find out what happens to these sets of patients that we treat. So you've heard so far regenerative medicine, stem cell therapy, etc. One of the things we really want to know, as well as the medical outcomes for these patients, is what happens to them. Do they still go to school? Do they? What happens to them in other admissions? What other data do we have available about them? So that's one key element for us. You're also then going to hear from Stuart and Rossa, who are going to show you how we can make better decisions from the data that we have from previous patients to make better decisions for future patients. And then we're going to come on to one of the innovation sessions afterwards. But I think there is a short video to whet your appetite. The aim of the Applied Child Health Informatics theme is to make best use of the data that we have on children with rare and complex disorders so that we can manage them better in the future. We have cutting-edge infrastructure within the organisation where we can collate, de-identify, manage and analyse all of this data in a secure and safe way. So through our digital research environment structure, where we can utilise all of this data, we aim to benefit patients in two ways. Firstly, by supporting the speed and the accuracy of which we can do research across all of the BRC themes. And secondly, a direct impact on patients for how we can use the data that we hold on patients who have been to the hospital previously to better manage patients that we see in the future.
Okay. So another key part of our theme is to use national data from hospitals and state schools across England that have been collected for other purposes. And we will use these data to look at how new treatments and management for children with rare or complex conditions impact children's overall health and education. And so, of course, we want these different strands of our work to combine seamlessly together.
So the theme will be collaborating with other parts of the NIHR infrastructure. For example, we're working with the UCLH BRC specifically to look at the life course of data, so how children's data and adults with the same diseases may differ. And similarly, we're also working with other organisations such as Sheffield Children's Hospital through the NIHR Children and Young People's MedTech Cooperative, partly to look at device data in this paediatric setting, and also, importantly, working with children and young people themselves around how they feel about their data being used to develop new treatments and areas of management. Right, enough of that, and on to some real examples. So over to you, Kate.
Okay, I'll just hold this. Let me know if you can't hear me. Okay, so I'm just going to present an example of the type of work that's going to be done under the Applied Child Health Informatics theme. So just some quick definitions. I'm sure most of you in this room know this, but by major congenital anomalies, we use the CDC definition, which is abnormalities of structure or function of prenatal origin that have significant medical, social, or cosmetic consequences for the affected individual and typically require medical intervention, with some more common examples listed here. Individually, these conditions are quite rare, but altogether are thought to affect about 1 in every 45 births, according to registry data. Nowadays, about 95% of children with major congenital anomalies survive to primary school age, with thanks to improvements in early intervention. So they're more likely to go to school and require additional support with learning, which is where special educational needs provision comes in. So by SEND provision, I'm simply talking about any educational provision that's additional to or different from that given to other children of the same age. In England, we have two broad categories of SEND provision. So we've got something called SEND support, which is arranged by schools and includes additional classroom support, including some of the examples listed here. And we've also got EHCPs, and these are arranged by local authorities for children whose needs cannot be met by the lower level of provision. In this study, we also separate out specialist school attendance, which includes special schools and alternative provision. So the aim of this work is just a descriptive study to estimate the prevalence of special educational needs provision amongst children with major congenital anomalies born in England. So why is this important? Well, families and clinicians want prognostic information about children with specific conditions, and this type of information can be used for early service planning and for evaluating delivery too. To date, evidence has been limited to regional studies and based on a few conditions. So we've not been able to look at a multitude of conditions in this way. So the data source. So in this work, we use ACHI, which is the first database of linked education and health records in England. And I'll go through the different data sources used. So we start with hospital episode statistics. So this is statistics on all admissions to hospital in England. In this study, we look at births from 2003 to 2013. We can also use this data to follow children up to the age of 1 to define major congenital anomalies, and we do this using diagnostic codes based on an internationally recognised code list designed by EuroCat. So to this, we have linked national pupil database, and these are school censuses. And this is information on all state-funded primary school attendances in England. We actually, in ACHI, have data that goes up to 25 years of age, which shows the sort of the longitudinal nature of this data. In this study, we follow children up until before the start of COVID or until they reach the end of primary school, so about age 10 or 11. We also have linked in Get Information About Schools data, which is an open source data provided by the Department for Education, to find information about school type. So our final cohort, in this case, is 5.2 million children, of which we identify about 3.5% with any major congenital anomaly. So that's 181,000 children, so it really shows the value of this data. So just some results here. So this is quite a complex chart, so I'll just start over in the corner. So this is the prevalence of recorded SEND provision in primary school. So simply the bars represent the percentage of children that have any sort of SEND provision given to them in primary school. So children, if we start over here, children with no major congenital anomaly, about 24% of them get recorded SEND provision in primary school compared to 42% of children with any major congenital anomaly. Also in this work, we're able to look across 12 system-specific subgroups, and there's 25 individual conditions that we've looked at too. You'll see by the different colours here, these are the different levels of SEND provision that I've already described, and you can see that really the difference is at the higher level of SEND provision, where about 4 times children with any major congenital anomaly are about 4 times more likely to have recorded SEND provision. And I'll just pull out a couple more results. So you see that children with nervous system, eye and chromosomal anomalies have the highest prevalence of SEND provision across the groups, and again, this is really concentrated in the higher levels of need. For example, the second to last bar is Down syndrome, and nearly all children in this cohort with Down syndrome have recorded SEND provision in primary school. We also are able to look at children with isolated anomalies versus those with any, so across all system subgroups, and as expected, these children have a lower prevalence of recorded SEND. So just some key implications. So overall, we find that 2 in 5 children born in England with a major congenital anomaly have recorded SEND provision in primary school. And this starts to show the potential to use this de-identified data routinely for planning and monitoring SEND provision. But further validation of code lists is needed. It's also important to note that in this data, we have information about like a recording of SEND provision, but we don't know the exact provision that they're given, nor the quality of it. And there's work being done by Cambridge as part of the HOPE study, qualitative work to look at the impact by talking to families and children. Overall, we find quite a bit of variation amongst children with different conditions in this data, but we're also investigating other sources of variation in recorded SEND provision. So, for example, as part of this work, we've looked at changes that happened before and after a major policy shift, which happened in 2014-15, and we actually find that following this change, about 1 in 20 fewer children have SEND provision, which matches the broad level of unmet need as described by children and their families. We're also looking at variation across local authority, which we can do using these big data, and looking at things like the association with funding and type of governance of schools too. Thank you.
Brilliant, fantastic, Kate. You made that look very easy and straightforward, but we'll take questions at the end. But firstly, you can just see how powerful this could be for children with rare diseases, with novel therapies, etc. And secondly, I don't know any of you who have been involved in trying to do that kind of linkage. That one slide took 3-4 years to more, 5 years of approvals to do that linkage. So it's actually a massive task, so thank you very much. We'll take questions at the end, but Stuart, Rossa, over to you. So we're now going to give you an example of some of the kinds of things that can now be done with EPR data.
Hi everyone, thanks very much. My name's Stuart, I'm a data scientist in Drive in Gosh. So today I'm going to talk about some of the things that we can do with routinely acquired data that's already in the EPR. I wanted to start by just showing this slide, which is a summary of all the data that we have available in Gosh at the moment from the EPR and in previous administrative systems, to kind of show how much there is and yeah, how useful this could potentially be. You can see this is largely exponential for the last 20 years, and we've got a total of around 600 million data elements in the system, which can be used for different things. Obviously, these data have lots of primary uses in the clinical care of patients, but there's a whole load of other things that we can do with that data to kind of improve the patient. So obviously clinical research is a big area, but then also we can use that data to improve the clinical care of wider patients by aggregating data across interesting patient groups. We could look at sharing this data externally, so we've got obviously lots of information at Gosh about conditions that are not known elsewhere, which we could use. And there's a few other things here that we could do with this data. So for anyone who doesn't know, so the DRE is a group in Drive who have this amazing system whereby we can extract all of this information I showed you on the previous slide. And we've got 20 years worth of data, which they can extract, put into a common data model so it all looks nice and it's in the same format, de-identify it and then push it into these workspaces, a trusted research environment, so you can do analysis and you can do all these things really easily and very efficiently. So this is some work that's been going on for several years. And what we're trying to do now is build on top of this what we call a clinical analytics platform. And that's kind of taking that data and making it even more usable and accessible to researchers and also clinicians in the hospital. This slide here just shows the kind of the cycle of how clinical analytics things could potentially work. So the idea here is that we have a user or a clinician or someone who wants to know an answer about their data. They will request an extraction, so which is what the DRE do. We extract all that data from the EPR in a nice format. We do define some cohorts and have phenotypes of whatever your patient group of interest, and we can extract that. Bit of data preparation, obviously, to try and make things nice, and then build an analysis. Based on that analysis, we can then present the results back to the user in an interactive form, whether a dashboard or a report. And then ideally, this is the kind of crux of it, all of that work that we've done for one type of analysis we can save and then reuse for similar analyses in the future. And this core idea here is the basis of picture that we can generalize analytics and apply them across EPR data. So I'm going to hand over to Rossa now, who's going to explain how Picture, which is the platform we've built, can try and answer some questions for a clinical case.
Thanks, Stuart. Hi, I'm Rossa. I'm a respiratory and lung transplant consultant. Stuart built this super cool tool, and he just needed a blank canvas. I am that blank canvas. And I walked straight into his trap. These are the slides I sent Stuart originally, who's a data scientist. So because we're going to, some of the labels are a bit more, this is what's going on here. Don't laugh too much when I start talking about them. And so I walked into your trap in the middle of winter, and we were beginning to see quite a few group A strep cases coming to PRC with really bad empyema. And we've done a very boring, very traditional, very all collaborative hospitals. We've all got together and we've shared our cases and we've written them up and they've been submitted for publication. But there's no sort of, the information I find when parents come into hospital is they sort of believe the medicine is going to work and they sort of trust us, right? But the stuff I find that they're quite interested in is, what on earth am I going to do with my other kids while I'm here in PICU? How long am I going to be here for? And what's the kind of stuff that could happen to my child? So this tool, I think, is fantastic for giving us those sorts of insights. So this is a 2-year-old girl who I've plucked out of the sky, who I saw in clinic a couple of weeks ago. She had a bad empyema, and I was drawing for Stuart what that meant, which is essentially a large bag of pus sitting on the outside of her right lung there, and then the surgeons put a big pipe in it. And this has more complicated names, but pleurodesis fats, blah, 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 is a bit more boring. So she gets better. And we do the X-ray once afterwards. Don't do it too soon because the child always looks better than the X-ray. But after about a year, that is all looking fine. Dr. Stevenson at the back line, there's a little bit of pleural thickening at the right base because he did a big trial on this, but I'm going to ignore the fact that he's in the room. But the useful thing here, listen, don't concentrate on the numbers. The thing here is the cool stuff you can do with this. So the number of kids we had who came in with group A strep was not massive, but we can build this cohort using this tool. So the number of children who came in with empyema, pus in the chest, without group A strep and with group A strep, we can just chuck cohorts together and you can see the numbers unless you're at the back, which is we've got about 189 patients. Most of them don't have group A strep. Where's the numbers gone there? So 131 do and don't. And we can just build to this massive data set of lots of children who didn't have group A strep from years and years and years of GOSH data versus the kids in the last year who did. And we can see 7 had empyema with group A strep and 216 I think without, so we can see how many admissions that is and how many patients it is, the gender, ethnicity, the age they come into the cohort. Stuart hasn't included this, but he's got a very cool map of where they all live, which you probably shouldn't set up on film. And then you can do some various breakdowns, such as the demographics of, what does that say, the fractions of cohort? Why is there not like the men have to run across the stage? Male and female. Oh yeah, that's easy enough. And then you can look at other things that are a bit more useful, such as the ones who had group A strep and the ones who didn't. So if you had group A strep, you're in the red list, and this is the number, how old you were. Interestingly, they're a bit older. I don't wonder if that's something vaccines, really. Anyway. Then we can do how long you stayed in hospital. This is where things are beginning to get interesting. So the group A strep kids are sicker. We all thought that, but here's the proof. We've got a probability distribution that's showing you just how long these children are in hospital. In PICU, how long they're in hospital in total, the number of days of IVs you would sort of expect, because as soon as they're off IVs, we kick them out. And then this is my one that I thought the most interesting, the number of days of temperature. So these kids are sicker because they keep having fevers because we're struggling to shed this really nasty invasive infection. Don't concentrate on the numbers, it's like 5 kids. Concentrate on the kind of cool things you could do with this. If it's, I've been admitted with systemic JIA, how long am I going to be in hospital for? How many biologicals will I need? What do I look like compared to the cohort in this hospital or the hospitals we're paired with? So this is why Picture is such a cool tool. Come to us with your ideas for how we can make you pretty graphs. Stuart.
There was just one final thing, was to just highlight if there's any some of the other things that we're interested in doing. So one of them is obviously we've got this amazing access to all the data, and we can kind of deploy machine learning and AI things, which is what we're focused on, and then for clinical decision support, but then also integrating some of the other information that people here are probably interested in for genetics or novel treatments and therapies. And that's it. Thank you.
Any questions? Hopefully that's just whetted your appetite for the potential of the kinds of things we'll be able to do.
A couple of questions on Slido. Is this one? Someone's here.
Hello, this is a question for Dr. Kate Lewis. Hi. I've written it down because I know I'd forget. I just wondered if your inclination was potentially that the higher prevalence of the SEN support was related to educational professionals or associated agencies like local authorities, their awareness of chromosomal disorders versus the isolated MCAs that you talked about, and relatedly whether the support in place was influenced by the sample size of across those two different systems. Sorry, I'm not being very articulate, but were there more in your data set that had chromosomal disorders versus the kind of the other one that you talked about?
Yeah, sure. That one. So there are more children in the data set. The biggest numbers are with heart defects. It broadly matches what you'll find in any sort of prevalence study. I think there's about 5,000 children with Down syndrome roughly, or 6,000, off the top of my head. It's a good point. I can't answer that, whether you think it's influenced, whether I think it's influenced by the fact that I guess a child with Down syndrome is obviously presenting with Down syndrome, but then also they probably are more likely to have intellectual disability comorbid with Down syndrome. I mean, I think 100% of children do. So there's a lot to unpick in that information. I think it gives a good prognostic tool, but understanding the extent to which it is like fair for those children to have that SEN support or not is a harder question to answer. And it is something we're doing. We're employing like quasi experimental methods by using sort of different instruments to be able to figure out whether that is actually fair or not. But it's a good question. Yeah. Thank you.
Thank you very much. Amazing what you're showing, and you're talking about AI and machine learning, and obviously things that can be done. But at the same time, we know now there's the warning signs about using AI, and they may take over things. So do you have any protection so that the AI is not going to start prescribing antibiotics and putting chest drains in?
So the kind of things that we're thinking about at the moment are very limited in what we're considering allowing it to do. And I think we're a long way away from that. At the moment, the platform is very much focused on describing things and trying to communicate that to a clinician, but because of the way we've built it, we have access to all the things that you would need to do AI deployment. It's an aspiration rather than somewhere.
It is interesting to point more about how we are looking already at kind of MHRA regulation for the various elements, because there are different grades of what it's okay for a tool to do.
Thank you very much for both the talks. Picture looks amazing. As somebody who has spent a lot of time researching electronic patient records to retrieve data, do you think there'll come a point that you can use it also for perspective and characterization of cohorts? Because obviously that's also something that will add value to the data collection itself and to prospectively describe the cohorts. And then the second thing is, there's a limitation to this, which is clinicians, we are usually very bad at inputting data, as you well know. And how do you think that can be bridged with these AI technologies so that we can understand each other in what you look at as relevant data? Thank you.
Okay, yeah, 2 points there. So the second one first. I sort of have an idea we will eventually end up with an informatics-based ward round, where we will go around with patients with the computer on wheels, and the poor old SHO will be plugging these numbers into the picture when the patients ask a simple question like, how long before the pumps get delivered to my community care team so I can go home with my PEG JV, which is one of the major reasons kids sit in a hospital, certainly in my ward. And we go bang, 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 we can tell them, and we can show that data to commissioners and say, this is how slow things are going. From an operational side, it's really powerful. And to get the good data in, to get the good data out, we're going to need evidence-based informatics energized doctors, nurses, AHPs. I think those people exist in this building, and then we can use that as an exemplar to roll out and show people. So this is kind of the best place to do this first. But I'm an extremely optimistic human being, which I read is good for my life chances, but sometimes I'm open to disappointment. We'll see. And the second question about prospective cohorts, that's a machine learning, unsupervised hierarchy sort of research side that we also have going on, which isn't in the talk today, but look out for adverts to be our PhD students and come and do this work with us. We're setting that sort of stuff up.
Thanks, that was great. Just a bit of a related question really about whether we could have patient captured data that's integrated with our clinician captured data, so whether we could be using, for us, seizure diaries, the bane of our life, seizure diaries or activity monitors or other ways of capturing more granular data that we can then incorporate with our own data.
I had a meeting about that this morning. The MyGosh blob server is a safeguarding in informatics, information governance. I'm not going to use any subjective words. It's a difficult area at the moment because patients can basically upload what they want. And pulling the themes out from that in a systematic way, I think, will be tricky. But yeah, for example, 1990s asthma clinics talk about peak flow diaries, and you'll get a time and date stamp on your upload, so it won't all be written in the same pen in the 20 minutes while they're waiting in the waiting room, and you'll know if that data is useful or not. So yeah, that is doable currently. It's just make sure the systems are correct, like the responsible AI, before we roll it out, and that will take massive GPIE. Because it'd be much better if patients tell us what these systems should look like and then they design it, and then we can just say fine and press go.
I'm just going to take one question from Slido that says, this is for Kate, so you're using SEMPROvision, but are you using any other educational statistics as surrogate markers to help?
Am I using any other?
So you're looking at SEMPROvision as a marker of health, etc.
Yeah.
But are there other markers? Are you looking at anything else other than educational, so attendance or anything?
Yeah, I see what you mean. So I think really we use SEMPROvision as a marker of whether they get given provision rather than a marker of them needing it, if that makes sense. So we're really trying to there's Anya Zilberstein who works with us is really defining the code list better, so we're really focused on kids that should get SEMPROvision. But we are also looking at absences, which are obviously very important amongst these groups of children, exclusions and attainment as well as we can also obviously look at hospital records and admissions over time.
Brilliant, thank you very much, all speakers. And now for something not quite completely different, but a bit different, because an academic surgeon and a data scientist to show how we can start to bridge data with other aspects of clinical. So over to you, Stavros and George.
Good afternoon, everyone. Good afternoon, everybody. My name is Stavros. I'm an academic pediatric surgeon, and I'm a new PI at the Institute of Child Health. And this is...
Hi, I'm George Hall. I'm a postdoctoral bioinformatician. I work in Sergi Castellano's lab across a number of BRC-funded projects.
And really the purpose of this presentation is to show you how the BRC has brought myself and George and quite a few other people together to try and do something that has to do nothing with surgery. So I'll talk to you a little bit about how we've been using BRC support and funds to unlock the hematopoietic potential of amniotic fluid stem cells for therapy before birth. And the first, so my group and quite a few others, both here at UCLH, were trying to develop therapies for babies before they are born. I'm sure you have heard about surgery for spina bifida and the fetoscopic tracheal occlusion for congenital traumatic hernia. But we are looking into something that's a little bit more generalizable. So what's the rationale? Why are we trying to treat sick babies before birth? Well, we are targeting diseases that cannot be cured after birth. And we're trying to provide treatment before permanent damage develops. There's a few things that are developing during pregnancy that we cannot fix after birth. The fetal environment offers you access to stem cell niches that are not as accessible after birth. And we have the ability, due to the small size of the fetus, to administer large treatment doses relative to weight. The development of immune tolerance is also an important thing. The fetus is immunologically mature, so you can transplant cells across immune barriers without a requirement for immunosuppression or any toxic treatment. And then finally, as a clinician, it's important for me to be able to give parents a choice other than termination or long-term disability. Now, my group and our group focuses on inherited hematological disorders, in particular for alpha thalassemia major. And the indication for that is that alpha thalassemia major has a fetal phenotype with the development of heart failure, hydrops fetalis, and a fetal demise. So one of the indications for fetal therapy, and one of the clear indications of fetal therapy right now, apart from development of injury that cannot be treated after birth, is fetal demise. And we have discussed, with the support of the BRC, with quite a few of the relevant patient groups, and there's quite a lot of support for the development of novel therapies, mostly due to the fact that the therapies we have at the moment, they're not as good as they should be. And we have two options. The first option, which is the gold standard option for fetal therapy at the moment, is to replace the baby's defective stem cells with healthy stem cells from an adult donor. And the way this happens is you do a bone marrow harvest like you would do for postnatal therapy. You don't require any matching, so you can take whatever bone marrow is available. You can process it, store it, and then you can transplant it before birth, but you have to do this during the early stages of gestation. The benefits, the advantages of this treatment is that you can transplant allogeneic stem cells without matching. As I told you before, immunological maturity is one of the benefits of fetal therapy, and you don't have a requirement for conditioning, wiping out the bone marrow, and immunosuppression, suppressing the bone marrow in order not to reject the cells. Now, the challenge is mostly that fetal stem cells outcompete adult equivalents for space and niche. Mice stem cells and fetus stem cells, they're not quite the same. The fetus is programmed to proliferate, enlarge, so mice stem cells will not have the same competitive advantage as the fetal stem cells. And more importantly, and that's a technical and risk problem, there's a requirement for early gestation transplantation prior to fetal immunological maturation. So I have to go in during early gestation, and as a result, this creates risks and technical problems. That's why we started working on the second option, which is to replace the baby's defective stem cells with baby's own genetically corrected stem cells from the amniotic fluid. And this really mimics the postnatal autologous ex vivo gene correction and stem cell transplantation that is used clinically at the moment. So what we propose is amniocentesis, which happens really at around 16 weeks of gestation, isolation of the amniotic fluid stem cells, ex vivo gene engineering or therapy, and then expansion and then transplantation before birth. And you can do this much later because these are autologous cells, so the immunological issues that you have, they're not there anymore. It's very important to understand that one of the key things that we have is expansion. Amniotic fluid stem cells, there are quite a small number of those, and you need to expand them in order to be able to get therapeutically relevant numbers of cells. So I've discussed with you about the advantages. They are autologous. There are cells of fetal origins with favorable competitive characteristics, and there is reduced risk of insertional mutagenesis and no risk of germinal modification because you're doing all your gene therapy in the dish. Now the challenges, when we started working on this, the in vivo hematopoietic activity of those cells was not demonstrated conclusively, and there was requirement for expansion without the loss of hematopoietic potential. And I have to tell you that this has been something that I've been trying to do for a little while. So amniotic fluid stem cells were first introduced by Paolo De Coppi, who is a mentor and a colleague, pediatric surgery colleague. He described them during his PhD in the United States. And essentially, they are isolated by using CD117 selection, expansion, and then they can be differentiated in all lineages, endoderm, mesoderm, and ectoderm, without the ability to form any tumors. So the term broadly multipotent has become for them due to this fact. Now, a paper that was published in 2009, sort of soon after the first description, demonstrated that amniotic fluid stem cells, the CD117 portion of amniotic fluid stem cells, have hematopoietic potential. You can see that they express CD45. They can form colonies in vitro, and that's both in human and in murine cells. So human freshly isolated CD117 positive amniotic fluid stem cells express hematopoietic markers, but those hematopoietic markers are lost when CD117 positive cells are cultured in adherence. So we cannot really use the culture methods that we've had at the moment in order to maintain and expand those cells. Now, Following this, and this is where sort of my postdoctoral work and the focus of my lab is, we tried to translate, to do some experiments and demonstrate the utility of those cells in the in utero setting. And this is the first publication that was stem cell development in 2018 with my colleague Panicos Sangaris, who is now a fetal medicine specialist at King's. And we essentially demonstrated the feasibility of this, the fact that you can isolate the fresh amniotic fluid stem cells, you can put them in a mouse in utero, and you can get levels of engraftment that are significantly over and above what you get with adult cells. Engraftment is multi-lineage, so all bloodlines, and there's no evidence of any tumor transformation. And then in a second paper, a year later, we demonstrated the importance of culture, and we showed that you can isolate amniotic fluid stem cells, you can put them on feeder layers, expand them, and you can maintain the hematopoietic characteristics without loss. And we demonstrated by administering those cells intravenously in the fetal circulation that you can achieve levels of engraftment of approximately 20% or above with just a very small number of cells. And the 20% is a gold standard because this will give you treatment and cure for diseases like alpha thalassemia. So we've been working along this sort of grid with, on the y-axis, you've got the hematopoietic populated capacity, and on the x-axis, terminal differentiation. And we feel that fresh amniotic fluid stem cells are sitting somewhere there. Culture pushes them closer, your fetal liver HSCs. And it looks like if you put them in the appropriate environment, you may be getting some sort of inhibitory programming. And the reason why we think that is because when we look at the gene profile of those cells, they're not quite hematopoietic stem cells, but they're not quite embryonic stem cells. There seems to be something in the middle. So following this, one of the first things that we did was to try and demonstrate in vivo the hematopoietic potential of freshly isolated amniotic fluid stem cells. And what you can see here is a comparison of those cells with cord blood HSCs. And essentially what we demonstrated with quite a lot of difficulty, because we had to isolate sample after sample and cell after cell, that cord blood HSCs and amniotic fluid stem cells, when you transplant them in an NSG mouse, they perform pretty much the same. And we confirmed the in vivo hematopoietic ability of our cells, both in primary and secondary transplantation. However, I then spent the next 3 years of my life trying to expand those cells, and I failed to do so. And I couldn't understand why, and almost gave up really doing that. And this is where the BRC came in. And the BRC came in two different ways. Firstly, it supported this gentleman and his group and our group were working together to develop a sorting strategy to only feed living cells to our single cell analysis system. So we have achieved that by ignoring FCS and SCS. We used HUGs to isolate nucleated cells and PI to exclude dead and damaged cells. And then essentially, we obtained a population that was sorted but are purified. And this then allowed us to work with people like George, Sergey, and other people within the Institute and the BRC to perform single cell analysis to try and see, well, where are those hematopoietic stem cells in the amniotic fluid? George?
Thank you. So I'm going to quickly try and talk us through some of the single cell transcriptomic work that we've carried out so far in this project. So this is what a single cell transcriptomic data set looks like. So each point in this what's called a UMAP here represents a cell. Cells cluster more closely together, the more similar their transcriptomes are. And I've colored each cell here according to the sample of origin. So this is what I've called the initial UMAP. This is with no cleaning up of the data set, and we have some sources of contamination that we need to remove before we can start trying to identify the stem cells within this data set. So the first thing we need to do is eliminate any maternal contamination that we may have picked up during the sampling process. And we've done so using globin expression. So very briefly, during gestation, we have something called gamma globin, which is more highly expressed than beta globin. And then following birth, this expression flips, and beta globin is more highly expressed than gamma. And if we look at the two sort of plot showing the expression of these two types of globin, in our data set, we can see that HBB, the beta globin, is highly expressed in this cluster of cells up here. And the gamma globin is not very highly expressed in this cluster, so this leads us to think that these are maternal contaminants, so we can remove them from our data. So when we've done that, we've slightly moved towards a cleaner data set, but we still have some cell types that we don't really want to focus on in this analysis. So we really only want hematopoietic cells in our data set. We have this big chunk of cells here, which drew our attention, and we wondered what they were. And we found that they express high levels of markers of epithelial cells. So again, we can basically come to the conclusion that these are contaminants, not this time of maternal cells, but of epithelial cells that we're not too interested in this part of the analysis. So again, we can get rid of them. We're getting closer now to a cleaner data set, but we still, before we can look for the stem cells, we need to do one more bit of cleaning. Your attention is maybe drawn on the right of this sample that's colored in pink, and it's showing characteristics basically indicative of low quality in single cell data. So we remove this sample along with a couple of other samples that also perform poorly on some more sophisticated quality control metrics. See if that works. No. It's gone. See if I can play on here. Hey, great. Okay, so we've ended up with our clean data set. This is what, it's not worked. There we go. I'll move to this. So we ended up with these cell types, basically. We've annotated cell types using the canonical markers for different hematopoietic cells once we've removed the contaminants. So then we can begin looking for the hematopoietic stem cells. We've done so using this recent Nature paper by the lab of Hannah Mikkola in San Diego. And we've been very fortunate that the first author of this, Vincenzo Carbonesi, has moved to UCL. So we've been working now in collaboration with him to use his expertise to look for the hematopoietic stem cells in our data. And what we've learned from this lab is that we can use the co-expression of these two genes, RUNX1 and HLF, to aid us in our identification. So RUNX1 shows us where the hematopoietic cells are, and then HLF shows us within the hematopoietic cells the location of the stem cells. How does this look in our data set? Well, as you might expect, the marker for the hematopoietic cells is largely expressed throughout. Within the cells that express RUNX1, there's a subset on the right that express HLF. So this is our cluster that we're, that I've colored in yellow here, that I'm considering as being HSC-like. So they express a phenotype similar to HSCs. Going a bit further, I've got a histogram here of what I call the differentiation potential of each cell. So I've done this by looking at effectively how committed to a certain differentiation lineage each cell's transcriptome appears to be. And the cells in yellow are generally at the higher end of this differentiation potential scale compared to the larger population in blue. So there is, in some sense, an upregulation of differentiation potential in the cells that we believe to be HSC-like. Further, we can infer a differentiation trajectory across this data set, and we find some branching differentiation trajectories starting out in our HSC-like cells and moving through the rest of the hematopoietic cells. So again, this is indicating that there's probably some stem-like features in our cells on the right of this UMAP.
So, thank you, George. So essentially, we would like to close by saying that we feel that within the SIGI population that you can see there, we have managed to identify some cells that express a hematopoietic stem cell signature based on Vincenzo Calvanese, who is now a collaborator in our work. The BRC has brought myself and George together. There was no way, without George's expertise, I would be able to actually do all this, and credit all to you and your team for actually helping us. But we feel that the BRC has helped us identify those cells. The BRC is still helping us continue this quest and try to find specific markers for us to be able to pick those out in a more reliable way. And the BRC has actually helped me to try and achieve something I've been trying to do for at least 5 or 6 years, which is to try and expand those cells, to try and use them in a patient before birth. Cells are now growing in culture for the first time, and we're really excited to be able to achieve that. The BRCs continue to support us in that, and I would like to stop here and say thank you very much for your help. It's not working anymore. There's a last box that needs to come in for the thank yous. There you go.
Brilliant, thank you very much. Awesome work. The single cell work, along with everything else, is just fantastic how it brings it all together. We're quite short of time, but is there any burning question? Okay. Just because we're short of time, we're going to continue on, but thank you very much. Super impressive work, and we look forward to seeing the next phase and seeing the first patient treated. Thank you very much.